Hello and welcome everyone to this podcast for genetics. In the last podcast, we began talking about gene regulation, and then we spent quite a bit of time talking about how bacteria regulate their genes in these structures called operons. In this podcast, I want to talk very specifically about the lactose operon, which from here on we'll just call the Lac Operon. To begin talking about the Lac Operon, I want to talk about two French scientists called Jacob and Manob. In their work and other people's work, they discovered that if you took E. coli and you plated them on a plate here that only contained glucose, they would grow and of course survive. They also knew if you took these same E. coli and you plated them on a petri dish that only contained lactose, that they would also grow and survive. Next, they plated them on media that contained glucose and lactose. They also grew and survived. So at this point, this doesn't sound all that interesting. But what they discovered was that in this third plate, E. coli didn't just use either glucose or lactose. They used glucose first. They ignored the lactose until all the glucose was gone, and then they metabolized lactose. Well, why is that? Well, let's think about the structure of glucose. Glucose is a 6-carbon monosaccharide. So it's just one ring. Lactose, on the other hand, is a disaccharide that contains one glucose molecule attached to one galactose molecule. So they reasoned that this being a single monosaccharide, that the E. coli is going to use this first because it's easier to consume one molecule of glucose. For lactose, it's going to have to break this disaccharide down into the monosaccharide glucose before it can be used. So it's going to cost more energy to use this disaccharide. E. coli, being the efficient organisms that they are going to prefer to use the glucose molecule first. No reason to waste energy when you have the good sugar right there. They reasoned then that this would be a very good system to use to understand the Lac Operon. That operon in E. coli that makes enzymes necessary to break down lactose. One would assume that as long as glucose was present, the Lac Operon would be off. And as soon as glucose was all used up, the Lac Operon would be turned on. That is what they discovered, but let's first talk about the parts of the Lac Operon. So let's draw our friend here, the Lac Operon, put our promoter here, the operator here, and then we need to draw its 3 structural genes necessary to metabolize lactose. These 3 genes are called LAC-Z, LAC-Y, and LAC-A. Just like we talked about before, this will produce a messenger RNA that have the coding information for LAC-Z, Y, and A that will then go on to make proteins through translation called LAC-Z protein, LAC-Y protein, and LAC-A protein. Okay, so what do these 3 proteins do? In order to understand that a little bit better, over here I'm going to draw a large E. coli cell. Now usually lactose in the medium is out here, not inside the cell. The way it gets inside of the cell is using LAC-Y. LAC-Y makes a protein that we call permease. Permease makes a channel in the cell wall of E. coli, allowing lactose to enter into the cell. And remember, lactose is a disaccharide composed of one molecule of glucose and one molecule of galactose. Now, some of the lactose, when it enters the cell, is converted into something we call allolactose. Allolactose then regulates the Lac Operon. And we'll come back to that in just a moment. Most of the lactose that enters the cell is metabolized into glucose plus galactose. And how does that happen? LAC-Z gene here that makes the LAC-Z protein is something we call beta beta-galactosidase. This beta-galactosidase is an enzyme that breaks this disaccharide in half, making the glucose and galactose, which are two monosaccharides that can now be readily metabolized inside of the cell. For the purpose of this class, we're not going to talk about Lac A, except to say that it makes an enzyme called transacetylase. Make sure you know the function of gene Z and gene Y. Alright, now I'm going to erase a lot of this here on the bottom, because I want to talk about how we're going to regulate the Lac Operon. The Lac Operon, from what we talked about on the last podcast, would be described as an inducible operon, meaning that the Lac Operon is usually turned off because the E. coli cell is going to prefer to metabolize the glucose that is there first before it metabolizes lactose. So for that reason, usually these genes of the Lac Operon are off. How do we keep it off? Much like we saw in the last podcast, there is a gene here that codes for a regulator. And this first regulator that we're going to talk about is a repressor. In your textbook and in the literature, this is often called Lac I. So that's what we'll call it. Or sometimes we'll just call it I. So as we said before, it's inducible. So usually Lac I is going to be bound to the operator. And as long as Lac I is bound to the operator, RNA polymerase can't move past it. There is also an activator involved in this, but I don't want to talk about it yet. I want to talk about just the repressor for now. So in a cell where there's glucose present, the repressor, Lac I, will be made and it will bind to the operator, preventing transcription of these genes. Now what we see here describes a situation where the medium contains glucose. Glucose is present, the Lac I repressor is bound to the operator, preventing transcription of these genes necessary to metabolize lactose. But since there's no lactose there, it's not going to turn these genes on and waste that energy. Now, when glucose is absent and we add lactose to the medium with no glucose, it's important that there's no glucose here, because as long as there's a little bit of glucose, it's going to use that. So with lactose, no glucose. What happens is lactose that enters the cell is converted into allolactose. Allolactose serves as an inducer. So when allolactose is made, it will bind to the inhibitor, Lac I protein. When it does that, it causes a conformational change, and the I repressor is removed. So let's go ahead and do that. So now we have our I repressor here with the allolactose inducer bound to it. Once that happens, RNA polymerase can bind and turn these genes on. So now it can metabolize lactose into usable sugars for the cell. So again, when glucose is present, the repressor is bound to the operator. Once glucose is lost or gone and lactose is present, the inducer, allolactose, binds to the inhibitor and pulls it off the operator, allowing these genes to be transcribed. So this is half the story. We've talked about the repressor I and how that's involved in this inducible operon. But we haven't yet talked about the activator. So I want to spend some time talking about the activator. Because remember, to turn this operon fully on, we need to remove the inhibitor and add the activator. So let's talk about the activator. So remember, under normal circumstances, where there's glucose in this cell, in the media, the repressor is bound, the Lac I repressor is bound to the operator, and the activator is also not bound to the promoter. As we just saw before I erased it, when we remove glucose and add lactose, and again remember no glucose, the I repressor is removed, so I'm just going to erase that. It's no longer near here. And now we need to bring the activator to the promoter. So I'm going to give the activator a specific name now. The activator for the Lac Operon is called CAP. Once CAP binds to the promoter, it will recruit RNA polymerase and we can turn these genes on. However, CAP doesn't just naturally bind there. It also needs an inducer. So CAP, remember, which is the activator, plus an inducer, which we're going to call cyclic AMP. So once these two combine, and I'm going to draw them here with cap bound to the promoter with cyclic AMP attached to it. That serves as the activator. Now remember, we only want this to happen when no glucose is in the cell and there is lactose in the cell. The lactose helps remove the repressor. The lack of glucose is what helps make cyclic AMP. So we're going to explain how we make cyclic AMP only when glucose is absent. And to talk about that, I need to talk about ATP. ATP is converted into cyclic AMP by an enzyme called adenyl cyclase. So remember, we only want to make cyclic AMP when there's no glucose. But when there is glucose, we don't want to make cyclic AMP. And so what happens is, glucose, when it's in the cell, it inhibits this step here. So when glucose is in the cell, we make no cyclic AMP. It inhibits this enzyme, adenyl cyclase. ATP to cyclic AMP is blocked. When we lose glucose from the cell, meaning it's been all consumed, so there's no more glucose left, we can get rid of this here. And now adenyl cyclase is no longer inhibited. So now we can once again make cyclic AMP. When cyclic AMP is made, it will bind to cap. Once it binds to cap, the cap with cyclic AMP will bind to the promoter, recruiting RNA polymerase right here to begin to transcribe these genes, Z, Y, and A. Okay, what I'd like to do now is do a quick overview of the Lac Operon under various circumstances. Okay, in this first situation, let's consider a cell that has plus glucose and no lactose. In this situation, we're going to have our Lac Operon with our promoter, with our operator, and of course with our 3 open reading frames, Z, Y, A. If glucose is in the cell, that means cap, our activator, will not be bound. And remember, it's not bound because ATP cannot be converted into cyclic AMP because glucose prevents this from happening. So cap will not bind to the promoter because it doesn't have the inducer bound to it to activate it. In addition, the Lac-I repressor is bound to the operator because there's no lactose in the cell to pull it off from the operator. And as a result, RNA polymerase is just hanging out here. It cannot transcribe these genes, so this system is effectively off. And so you don't make these proteins. Now, what happens when we add lactose to the cell? We still have glucose in the cell, so we're still not going to make cyclic AMP. And if we're not making cyclic AMP, cap will still not be bound to the promoter. And RNA polymerase will not be very effective at binding to the promoter. Interestingly though, as long as lactose is in the cell, it can be converted into allolactose, and allolactose will bind to the inhibitor and pull it off the operator. So now we have our inhibitor with the allolactose attached to it. Now this alone will not be enough to turn this operon on because our cap is still not bound to it, our activator is still not bound to it. You will get some expression from here, but not a lot, just a little. Because in order to activate it, you have to have both that cap bound to it and the repressor off. So we're halfway there. Now after a certain amount of time, all this glucose is going to get used up. And once this glucose is used up, there's no glucose there, and so our enzyme adenyl cyclase is no longer inhibited, and we can make cyclic AMP. If we can make cyclic AMP, it can bind to the cap, and now cap can bind to the promoter with the inducer attached to it, and in doing so, recruits RNA polymerase to the promoter. And once RNA polymerase is to the promoter, and remember the repressor is pulled off because we have lactose in the cell, now we can fully turn these genes on. We can only fully turn this operon on when glucose is absent because that helps activate cap the activator, and when lactose is present because that helps inactivate or pull off the inhibitor. Now at some point what will happen is lactose will get all used up because Lac-Z has metabolized it all. And so now lactose will be reduced. And because lactose gets turned into glucose and galactose, glucose will be high again. And as a result, everything goes back in reverse. Allolactose is no longer present because we've used up all the lactose. Our inhibitor will now bind to the operator. And because glucose is present, it's going to inhibit cyclic AMP production again. And now the cap, once again, will fall off, turning off the operon. It can go back and forth in its expression of this operon based upon the availability of glucose and lactose. Okay, that's all I have for this podcast on the Lac Operon. In the next podcast, we're going to talk about various mutations that were found that helped them understand how the Lac Operon functioned. But if you have any questions over this material, please make sure you come see me. If not, I'll see you in class. Bye.